I'm going to talk to you about my experience in primary ciliary dyskinesia, and I'm going to start with a little bit about who I am and what gives me some credentials to do that for you. So I'm from the University of Southampton. I'm a senior research fellow in the Faculty of Medicine, and I'm also a member affiliated to our NIHR funded Biomedical Research Centre. But our centre is a hub for PCD diagnostic and management activity in the UK. So I've got 16 years of experience, starting from knowing nothing all the way through all the accrued methods to diagnose PCD in the laboratory, and all of the cutting edge advances that have occurred since genetics has come online in the past few sort of 10 years. So if you want to go and look at my back catalogue of publications, there are others that have published far more than me, but you can see what I'm about using, referring to my ORCID reference there. So I'm the diagnostic laboratory lead at Southampton, which means that I lead a team of technicians and other research specialists that perform some of the diagnostic tests that I started out developing. But I'm fundamentally an airway cell biologist, and I've used what I know when I can for outreach projects. So I've been developed as an engager mentor, which means that I've performed outreach tasks to engage the public and patients in what PCD is, but also I can help others to develop their outreach projects as well. And PCD Support UK is the patient, family and supporter run charity in the UK, and we do a lot of cross-interacting with those, as well as we're NHS funded and commissioned, so we meet regularly annually, and the scientists meet up at interim points in our annual service, which means that we can exchange and share protocols, we can make updates, find out when things are not going so well, or support each other with difficult cases. So as far as BEAT-PCD goes, I've been a member since it first started in 2012. It was Southampton led by Professor Jane Lucas, and I've been a trainer for some of the workshops that BEAT-PCD training schools have offered over the years. And if you'd like to get in touch with me, if there's anything that you see that I talk about, you're welcome to email me and make contact, and I'll see if I can help. So today's presentation is about sort of describing how we integrate, or how we're now integrating, genomic analysis into our clinical and molecular data, and how we're using this to stratify our PCD patients much better than we used to. I'm going to give you a little bit of history about PCD. There might be things that you already know and things that you don't find as useful, but I'm hoping that I demonstrate to you how we're taking PCD at the cutting edge front line into a new transcriptomic and genomic era. So I'm going to show you how we evaluate our genomic and transcriptomic data, and how we're using this to better assess whether certain PCD gene variants actually are pathogenic, because sometimes the functional data needs to fit more perfectly with the presumed responsible causative variants. And I also want to show you how this could potentially be used for the new molecular therapies that are being proposed as cures for PCD, which are so far lacking. So what do normal cilia look like? So this is an example of our best case high-speed video, where you can see a nice intact membrane surface. You can see a ball of epithelial cells lined with cilia beating in a coordinated manner and propelling particles in the liquid around them, demonstrating really effective mucosillary clearance. And when you perform this test, you have to try and standardize it as best you can. So you need to stabilize the pH, you need to stabilize the temperature, and all of the samples need to be processed in a very similar manner. We've got local data that allows us to understand what we consider as normal, and we also have built up we've built up experience in understanding which phenotypes we expect to see aligned to different ultrastructural defects. So here you can see an electron microscopy example of a healthy, normal motile cilia in cross-section, and it presents a 9 outer microtubule doublets, microtubules, and here on the leading microtubule you can see an outer and inner dining arm. You can see central microtubules surrounded by a sheath apparatus, and you can see some of these radial spoke proteins here. And so this is how we would represent normal cilia, and we expect there to be excellent mucociliary clearance. However, we do see some cases of PCD where mucociliary clearance still exists. So what is PCD? So it was first described in actually 1904. It was considered a combination of situs inversus. So you can see here that the body patterning is mirrored. So it's not the normal organ patterning. It's as if someone's held a mirror to this person. You can see they're all shifted to the other side. Chronic sinusitis and bronchiectasis, which is severe lung damage. And then Carter Gerner, who was a pulmonologist from Zurich, recognized that you could put bronchiectasis, situs inversus together, and it was considered to be a potential congenital syndrome. And then Afzelius recognized a connection between immotile cilia syndrome and male infertility, because you can see sperm tail ultrastructure mirrors that of airway cilia. But first estimates were considered to be that this is quite a rare disease, so something like 1 in 30 to 1 in 60,000 persons. And we've got new data that demonstrates that actually this is a huge underestimate. So what happens when we have impaired mucociliary clearance? So essentially you have a lack of mucus flow. The mucus is designed to trap particulates and pathogens and debris, and then the cilia in the periciliary fluid are designed to propel that up and out, up from the base of the lungs up to the mouth, that it could be expectorated or swallowed. And if this mucociliary clearance is impaired, then we can see a buildup of bacteria on the cell surface, and this can lead to biofilms and really recalcitrant infections. And so we find our patients have chronic infections from early on in life. And so this demonstrates here the different clinical symptoms that we expect to see in our patients, depending on, this is let's argue that this is an extremely, there's a spectrum of disease. So we've got some patients that are starting out their clinical journey and they're early on in life, and they haven't yet had time to accrue multiple infections. And so they don't necessarily have all of these symptoms, but they have things like neonatal respiratory distress, and they have an early wet cough that's on a daily basis, and possibly chronic rhinosinusitis symptoms. And then as they progress throughout life, they accrue hearing loss, and it leads to bronchiectasis, but this can occur from quite a young age. We've seen patients as young as 2, if not slightly younger, that have already acquired bronchiectasis. And then later in life, patients will find that they potentially have subfertility, but it's important to point out that not all PCD patients have infertility. And of course, laterality defects due to embryonic nodal cilia accruing a defect that causes a defect in left-right body patterning during the early signaling during development. This is something that you can pick up early, but there are many other reasons why laterality defects can occur. So this isn't unique to PCD. So this is the clinical picture that we see in our patients. And Before we did this study in 2014, others demonstrated that radio aerosol clearance could be one way of measuring how well a patient, or a difficult patient, potentially difficult to diagnose patient, was clearing radio aerosol from their lungs. So you can see on the top panel, we've got a healthy patient where they breathe in the radio labeled aerosol. Then after 1 hour, it starts to enter into the gut, and then more so after 4 hours, until it's completely cleared by 24 hours. But in this example of a PCD patient, you can see that the radio aerosol is retained in the lungs. You get very little gut clearance, and it's still there after 24 hours. So this was potentially touted as one way to diagnose extremely difficult to diagnose patients, but it's an extremely expensive, labor-intensive method that not all patients would tolerate. So this is a genetic disease. It's usually inherited as an autosomal recessive disorder. There are some rare X-linked cases. So we've got some examples here: RPGR, PIH1D3 and OFD1. So these are some of, so that's these two, retinitis pigmentosa and olfactory syndrome. These are a couple of different cases where it can be X-linked, but the majority are autosomal recessive inherited. So it has to come from both parents. There's a higher prevalence known to be in consanguineous families, where founding mutations are held within populations, and studies by O'Callaghan and others have shown that, for instance, in the UK population, it can be as prevalent as 1 in 2,000. And this was because they found in that study cohort that 52% of parents were first cousins. And so this is something that can be considered when you're looking at your demographics within your own study cohorts. So other ciliopathies to look out for: Bardet-Biedl syndrome, polycystic kidney, I can't say this, I can't say it. Excuse me. And then Alström syndrome, Meckel-Gruber, and then retinal degeneration. And if you look in your population, you're not going to pick these cases up very frequently, but every now and again, you're going to find syndromic cases where their cilia dysfunction isn't the more routine dysfunction that you note. So for instance, you get sparse cilia with that are extremely long with bulbous ends and things like that. It's worth considering which genetics panels that your patients are being filtered into for screening. So this is just to demonstrate that there are hubs of activity going on worldwide, and it's just to recognise that prevalence is really difficult to know until we know that PCD is being appropriately picked up and managed and properly screened for in all countries, and this at the moment isn't the case. So this is the initiative of BEAT-PCD, is to bring our networks together, and therefore we don't work in silos, and we can share information about our rare cohorts, and that we can increase activity between countries. So this is a study that has brought our understanding of prevalence a bit further along. So Will Hanna demonstrated that there was the global prevalence of PCD is much more frequent than we originally estimated, and that this was using specific genetic databases. And I just want to really highlight that Will produced a webinar that you might be interested to go and look at based on this publication. So what did he find? So they basically used 2 specific databases, and they catalogued the variants by primary ethnicity of the patients. Could everybody just make sure that they're on mute, just so I don't get some feedback? Thank you. And the minimum global prevalence is considered to be about 1 in 7,500 individuals. But if you look at the breakdown, so using the genome add allele frequency by ancestry, you can see that the higher prevalence tends to be in patients who have African descent. And if you were to include all the variants of uncertain pathogenicity, including all of the benign ones, you potentially see how frequent PCD could be. But we know that not all variants of uncertain significance are pathogenic. And this is a reason why our genetics has to improve and our variant detection and classification has to improve, so that we can really understand what our true prevalence is globally. And this also may vary by country and vary by demographic status. So if you look across the different ancestries, you can see that there are, this is looking at the top most commonest PCD genes. You can see that DNH11 and DNH5 come out most predominantly, but you can see there's a sort of an intermix of the other ones here labeled at the top and the mid of the screen. And it's basically, this isn't to go piece by piece through each major gene, but this is just to demonstrate to you that depending on ancestry, you can get a different profile. So this is just to acknowledge that the main founding genes in different countries might differ. So in the UK, we've had a national service since 2006. We've also developed a management service for children in 2012, but we've now developed a commissioned adult management service. So this is about developing diagnostics. This is about knowing what to do with our patients when we've diagnosed them. But the adult service that came online was really important, because this was allowing us to transition our child patients into adult patients care, which needed to be managed differently and required its own budget. So I work with partners in Leicester and the Royal Brompton, and we, as I've said already, we coordinate to make sure that our protocols are aligned and that we share our ideas and that we increase detection as much as possible. We also link out to networks of genetics labs. So Southampton links out to Wessex, but there are other genetics labs in the UK that act as hubs to provide genetic services for our centres. And then we try and piece the functional genetic data back together. So we conform to ERS task force guidelines that were set up. They were actually set up in 2016. So Jane Lucas led a task force that used evidence base to develop an advised diagnostic guideline for PCD. And this is just really to demonstrate the literature that you can go to look at this. So I've given you that newer literature from Amelia from 2019, because it's a comparison between the ERS and the ATS guideline, which do differ slightly. So the UK service subscribes to this method, which is about taking a history on a patient, performing nasal nitric oxide screening. But we don't stop there. We don't use nasal nitric oxide to prevent us going into other diagnostic tests. So we take nasal brushing from the patient and we perform high-speed video. And we aim to not have to repeat this test by carrying out cell culture protocols. And if we see abnormalities, we go on to carry out electron microscopy. And we also carry out genetic testing on most of our patients. If they have hallmark, I'll come on to this, but if they have hallmark electron microscopy defects in their ultrastructure, we can label our patients as positive. And if they have proven biallelic pathogenic or likely pathogenic mutations, and there's strong evidence for that functionally, they're also considered PCD positive. But if we have no ultrastructural defect and an unconfirmed genetics, we have a highly likely group here where they have abnormal high-speed video. They might have some TEM abnormalities that don't fall into a hallmark category. And so they're considered and treated as PCD. And it's only when all of these tests and the history is not that strong that we label patients as highly unlikely, but we never exclude PCD completely. So we say to patients that if symptoms continue or worsen, then they should revisit our service as best as possible. And at the moment, we're aiming to recall fewer patients if we can by having a diagnosis from first visit, but it is possible that patients come back 2 or 3 times for repeat visits so that we can accrue the data. For instance, in the case of high-speed video, at the moment, we advocate that on 3 different occasions. So what we do is we count high-speed video as at least a second occasion, and a third occasion would be a repeat visit. But we try to minimize this because we don't want the patients to suffer unnecessary additional brushings. So looking at the ATS guideline, there are some specific recommendations for onward testing. And it's possible that these are slightly too prescriptive, in my opinion. So they need to have these specific year-round daily cough beginning before 6 months of age, and year-round nasal congestion and organ laterality defects. And if the answer is no to this, they're considered PCD unlikely. And it is quite likely that there'll be some false negatives in here, particularly as I've mentioned that PCD isn't just one type of PCD, it's a very large spectrum of disease. So we're going to have some milder cases that are accruing heavier symptoms through life that wouldn't necessarily conform to this. So then obviously if there is nasal nitric oxide screening and it isn't particularly low, then it can also lead to potentially missed cases. So I guess the bottom line is there is a lot of similarity between the ATS and the ERS guideline in that genetics and electron microscopy can confirm a diagnosis. But there's potential to miss cases in our view. So we feel that the ERS guideline for the UK is less prescriptive, which allows us to have a lower threshold for including patients for testing. I know that there's a network of people out there that are looking to combine a prescribed a guideline for everyone to follow that's slightly more aligned to each other's opinions. I think my screen is, oh, there we go, I'm back on. Okay, so looking at our patient demographics, 74% of our patients present with neonatal respiratory distress, and a high proportion of them have rhinitis and persistent wet cough. But other symptoms that tend to be less frequent and perhaps accrue later in life are hearing loss and ear symptoms. But perhaps this is also to do with a lack of evidence that we're maybe as task forces and ENT specialists are engaging with PCD. This data will help us to spot these symptoms in our patients sooner. But because we know what symptoms to expect, it's allowed us to create a testing score, which is a predictive tool. So it does actually depend if the child has a daily wet cough. So if they don't have a daily wet cough, then the PICADR score won't work, and they don't. And this is something that needs to be considered, particularly for potentially mild cases. But if they do have a daily wet cough and you complete the score, the cutoff of 6 means that if they have a score greater than 6, then it's more likely that this patient has PCD. And if you combine this with nitric oxide testing, then this can be quite a strong indicator that that the person does have PCD after all. So if you look at, so this basically is a really nice review that will give you evidence of which genes to date have been detected, although it does still miss a few actually looking at this. and it demonstrates which genes relate to which component of the ultrastructure. So you can see we've got dyning arms, you've got docking complex, as well as structural proteins that make these. Also, you've got sort of radial spoke and nexin dyning regulatory complexes that are really important for coordinating motion, and also central pair. You tend to see more rotational phenotypes in patients that have central pair defects. But there's lots of other cytoplasmic assembly factors that are more difficult to provide functional evidence for. So I would encourage you to look at this paper to give you an idea of what genes we're looking at the moment. So also, I want to highlight that there's an international consensus guideline for electron microscopy. So I would encourage you to look at the ERJ publication from 2020. And what it does is it allows you to understand how to classify patient defects. So I won't go into it in detail, but basically it's about knowing how many cilia axonemes that you would have to count to statistically define whether a patient has a class 1 defect, such as a dining arm defect or an outer and inner dining arm defect, or a class 1B defect, which is inner dining arm with microtubular disorganization, although you do have to count more ciliary axonemes. And then a class 2 defect, it isn't a hallmark defect, but it can relate to central complex defects, mislocation of basal bodies, but you can have lower proportions of these defects. So hence why they're labeled a class 2 rather than a class 1 defect. But these are the ones that are more difficult to fully characterize. So if you go to publications in the literature and they say that a group of patients have a specific defect, but they don't highlight what proportion of axonemes were scored, this can potentially suggest that the evidence isn't necessarily correct, or that if they counted more axonemes, that then they would maybe disprove their own contention. So it's really important to look at this criteria and consider it alongside your own diagnostic routine. So because we have cases where let's argue that the electron microscopy appears to be fairly normal, there are different antibodies that we can employ to target specific proteins within a cilia structure, and it helps to indicate if that structure is defective or potentially absent. So if the protein is mislocalized or completely missing, as you can see in the right hand side of this screen here. So these are examples where the protein in red is present along the ciliary axoneme, although you can see some of the proteins don't necessarily go all the way along the axoneme. And you can see the cilia marker and you can see the overlay. This is what you expect to see. But in cases where the protein is absent, it can help to indicate that a whole structure has gone missing. And sometimes it can be functional evidence to uplift a variant of uncertain significance if you can show that a protein, a specific protein has gone missing. So in the example of DNH11, you would expect that the ciliary axoneme ultrastructure would look normal, but if that specific protein has gone missing, then it would help to indicate that it's a genuine DNH11 mutation. But what I need to tell you all is that sometimes proteins can continue to appear to be present. So you can apply an antibody and you can still detect the antigen, but it doesn't necessarily reflect that the patient who has got PCD but only has a bit of the protein generated, it's not going to show them up as abnormal. So there can be false positives, and false negatives would tend to occur if the sample quality was poor, if mucus was in the way, or if the antibody was failing to work. So always including a control in your staining run is something that I would highly recommend for every single run. So looking at these more difficult cases, there is actually an antibody called SPEF2 that associates with hiding in the outer sheath of the, so in this outer sheath of the central pair microtubules. So when hiding is, if you genetically acquire a hiding mutation, then you tend to also lose the structure that connects to SPEF2. So this was demonstrated in Heimer Onran's paper, and you can see that SPEF2 can be an extremely assistive immunofluorescent diagnostic for hiding mutation patients. So hiding has a pseudogene, which means that if you look genetically, sometimes you will pick up potential variants, but you won't know whether it's in the main hiding gene or the pseudogene, which doesn't produce a functional protein. So this is where this diagnostic test can be supportive. So if you've got a fairly normal looking ultrastructure, but you have abnormal or subtly abnormal high-speed video findings, adding this into a standard panel of immunofluorescent screening is really helpful. There's also new data coming out of the Royal Brompton. So they're using a computerized artificial intelligence learn system that allows to look at electron microscopy through, so let's argue it's no longer a person judging whether they can see a defect or not. It's a computer basically using it's learned evidence of what is completely normal to interrogate whether there's any differences in the sample it's being given to test. And there is potential to spot for absences in labeling here. So you can see that there's a deficit in, there's a deficit in labeling here, which you can see in the control, and this is something that the computer was able to spot using its artificial intelligence method. And so this rolling out in the future is one way in which we could interrogate our electron microscopy in a more automated fashion, and this could help us to spot some of our difficult cases. So I would encourage you to go and look at this work as well. So the majority of our PCD patients present with a really nice classical static cilia. And what's really helpful is that the electron microscopy is hallmark when you lose your outer and or inner dining arms. So, but this can be due to loss of structural so if you incur a structural dynin gene variant, it can also be if you've incurred a dynin pre-assembly factor variant. So although it's really easy to diagnose the patient, if you want to be able to genetically confirm what caused it, this can be quite tricky. So when you come to genetic analysis, sometimes it's very straightforward in that you have two variants inherited in trans, meaning that one comes on one allele from one parent and one is inherited on a different allele on the other allele from the other parent. And that's very straightforward when the variants are both considered to be pathogenic. But sometimes we only get one variant, and sometimes we have the other allele presents a VUS, and we don't know if this is pathogenic or benign. And then going into the functional evidence, you don't always easily tease out what has caused the static cilia and the loss of the dynin arm. Because if you show that DNH11 staining is absent, it's arguably because you've lost the whole structure. So you could also stain for DNH11 or DNA L1, and you will also see loss of those proteins as well. So the problem occurs is when you can't attribute the correct loss of protein to the gene, and this is where you can't functionally uplift certain variants of uncertain specificity. Right, so not all PCD cilia are motile, so you quite often get presented with scenes like this from high-speed video, but you can also see that there's debris, there's mucus, there's a lack of cell health, and sometimes some of these defects can be, that are still very severe, can actually be down to secondary defects. So this is a problem, and this is where cell culture can be used to renovate a sample, and you can reinterrogate it to see, well, if I go back to this screen, so there's different reasons for a genuine loss of coordination, and that could be due to central complex or microtubule defects, but this could be attributed to genes associated with axonemal rulers, nexin-dynein regulatory complexes, or radial spoke subunits. So there's loads of different potential variants that can cause this type of phenotype. So this is where cell culture can support. And if you use this QR code, you can go to and use this paper for the method. You can go and look at examples of pre and post culture videos. So you can see when PCD remains PCD and when secondary defects are renovated. Actually, I will also say that this is quite a timely method, an untimely method. So it can take up to 4 weeks to produce these cultures. But I must say that since using these cultures, it's really helped us, and particularly since 2018, it's really helped us to reduce our number of repeat patients. So we've looked at our trend data, and we can see that we get a reduction in recalls, and we get an increase in certainty of our diagnoses when the ALI is used. So although it's expensive and it requires expertise to perform it, I would really recommend this for specific cases that are quite difficult. So we have some cases that have transposition defects, and so they have this rotational phenotype. And this is because you get a loss of a central microtubule, and then you get one of the outer doublets twisting and moving into the center, which causes this classical rotation. The thing about the patient is that you will tend to find that they have normal situs, because the variants that are responsible for transposition defects don't affect embryonic nodal cilia, so their body patterning works out as expected. But what I will say about transposition defects is the percentage transposition defect you tend to find in your patients' samples is quite low. So it could be something that is potentially missed, and learning how to spot transposition defects is really important. So I'd definitely recommend going to the EM guideline. And if you're new to electron microscopy, I would definitely advise you to hook up with another centre that has experience, and see if you can go on a training mission and learn how to do the defect counting. We've definitely had people come to us for that specific reason. So looking at genes responsible for PCD in our UK cohorts, so we looked at just under 400 patients, and we can see in the UK that our most predominant mutations occur in DNH5 and then DNH11. So these are probably going to be easier to spot than DNH11, except because we do our high-speed video, they're extremely abnormal, so that's helpful. And then we get quite a few patients with CCDC 39 and 40, and also CCDC 103. So the CCDC 103 mutations are dynein assembly. So these ones in green, they associate with dynein assembly. And in yellow, it's with radial spoke and central complex assembly. Then we've got nexin-dynein regulatory complex assembly, and this is obviously all the structural dynein arm assembly. So what I would argue is that depending on your cohorts, not every cohort's going to have the exact same profile to the UK, but it's really good for you to have as your genetics comes online, as your diagnostic comes online, it's really good for you to try and profile your cohorts so that you can contribute to bigger studies. You'll note that this study only looked at 31 PCD genes, but that was at the time because that was the available number of genes screened on a specific DNA panel. But this is just to demonstrate our display of different genes in our cohort. So what did we do with that data? So we stratified our patients by gene grouping. So you could see dyne structure, dyne assembly, radial spoke, dyne regulatory complexes or other functions. And you can see that if you stratify it using a topological analysis, that the gene clusters group together quite nicely. So you can see like the orange yellow fit together and the green blue fit together. And then if you compare that to those specific groupings to their TEM results, you can see that it does. You can see that the dyne assembly definitely linked to inner and outer dyne arm defects, clustering, and that the central complex and the inner dyne arm with microtubule defects corresponded correctly to the radial spoke or central complex genes. And then if you looked at the high-speed video, you tended to find that the completely immotile or with weak residual movement also corresponded to the inner and outer dyne arm defects. So this fit the profile as well. And all your rotating ones were also in this grouping here. So there was a nice pattern falling out if we stratified our patients by gene grouping. And then using that gene grouping, we wanted to look at lung function and neonatal respiratory distress as clinical features of those patients. And we also wanted to look at which patients fell into different specific gene groups. So if you look here, you can see that the worst lung function in the dark blue here was attributed more so to the CCDC 39. And you can see that the best lung function was attributed more to the DNH11 cases. And then you can see these ones in the middle that were sort of had an intermediate poor lung function, but they also had slightly better lung function. It sort of horseshoes across here. They were also associated more so with DNH5 mutations. So what this topological analysis allowed us to do for the first time was genotype, phenotype study our patients and demonstrate that CCDC 39 patients really are, as we anecdotally expected, looking like they're symptomatically worse, whilst DNH11 patients tend to be milder. So looking at DNH11, so they do have an extremely abnormal cilia profile by high-speed video, but their ultrastructure appears to be quite normal by electron microscopy. So if you're a centre that's only relying on electron microscopy, these are cases that you could miss. And as I've just mentioned, because they're looking at their clinical profile, it appears that they're on the milder end of the spectrum. So these are patients that are coming into our service and they don't necessarily have the worst symptoms and they have a normal ultrastructure and they're at risk of being missed. And as you saw from our spread of patients by different gene, you saw that DNH11 is actually quite a high proportion of our patients. So what to do about them? There's a potential test that we can do called electron tomography, and it's a way of tilting the sample in a sort of, if you don't just look at it in one plane, you tilt the sample in different directions, and then you can accrue electron microscopy images across that tilted section, and it allows you to produce a 3D rendered computer image. So what this publication here, done by Amelia in 2018, helps to demonstrate is that you can use the volume of the overall microtubule, and then you can focus on the outer dining arm portion, and you can quantify the ratio of outer dining arm within the overall microtubule. And so if you look at healthy controls and PCD controls that don't have DNH11, you can see that they have a similar percentage of outer dining arm overlap to overall microtubule volume, but that the DNH11 cases fall below this, and this is significant. And then if you look at the 3D rendered image, you can actually see that here's an example of a normal outer dining arm structure bolted onto what would be one of the 9 outer microtubule doublets. You can see there's a hole. And it's the problem with tomography in some ways is that it's how the expert thresholds the sample to be able to reveal the hole without going too far, that they artificially introduce a loss of volume. So this is a good method that could be used. The only problem is that it's labor intensive, it requires expertise, it's extremely time consuming, but there is potential for it to be used for a difficult case, which could make all the difference for elevating a variant of uncertain pathogenicity. So at Southampton, we've also looked at whole genomes. So we know that we expect from the literature that between 1 and 13% of bronchiectasis adults actually have PCD. One of the issues of new gene detection is that 60% of variants are actually orphaned single families. And because there are over 50 genes encoding mutations that are either non-coding synonymous or missense mutations or structural variants, those are the ones that we can detect, but it's the 30% that we're not detecting that we're quite interested in picking up now. We think that so Mendelian disorders, there are over 6,000 rare diseases, and if you look at the genomes of some of these patients, and then you use transcriptomic analysis, we can find potentially a genetic uplift in those patients because they actually have splicing variants, and they also have intronic variants that have a significant effect on proteins. And so I'll come on to transcriptomics slightly later, but I'll tell you the outcome of the whole genome sequencing project that took place quite recently. So variants that came out of the sequencing project were tiered and filtered. So one of the criteria for tiering was that the variant that was detected by whole genome sequencing had to be considered rare in a healthy population. It had to be protein altering in at least one of the transcripts. Remember, we're looking at biallelic inheritance for the majority of PCD cases. And it needed to segregate appropriately. So for instance, if you inherit 2 mutations, but they're in cis, i.e. they're on the same allele coming from one parent, that doesn't mean that it's PCD. So the inheritance has to be that mutations are inherited in trans. So you've got, if it's a heterozygous biallelic pathogenic mutation, you'll get one on one allele and the other one on the other. So in order to tier the patients, they needed to fit this criteria. And then we had a group of patients that had very definite protein truncation, either caused by a stop gain mutation or a frame shift, or a splice donor or acceptor site that actually caused a protein, a deleterious effect on the protein. But there was a tier 2 where it tended to occur with missense mutations or splicing region mutations that was even though we understood that there was a potential functional problem, it wasn't as easy to consider it a pathogenic mutation. And then anyone that didn't fall into this using the panel app gene panel were tiered tier 3. So I would encourage you to look at this paper for the approach. And this is a short slide on the outcome, the main outcomes. So what did we find? So if you looked at 21 probands, so 21 genomes from 21 patients considered to have PCD, what this whole genome sequencing did was it confirmed 16 of them had confirmed probable or possible PCD because the mutations fell into place. But 13 of these should have been detected by standard gene panels. So all of these probands filtered into this 100,000 genome because they didn't have mutations picked up through standard methods. So what this affirmed to us is that some variants were being missed by standard panel testing. We also looked at a non-CF bronchiectasis cohort, and we found PCD in 3 of 52 probands. So probands again being patients, we looked at the patients' genomes, and 3 of them fit into confirmed, probable or possible PCD categories. But the strength of using this approach, apart from the fact that we managed to get a diagnosis in these 16 and these 3 patients, we actually identified for new PCD candidate variants. And this just basically here, this pie chart is just demonstrating again what I've already showed you, that DNH11, DNH5 and DNAI1 for outer arms tended to be the predominant variants found in all confirmed PCD cases. So what did we do with this information? So we suspected CEP164 was potentially causing PCD, and we've managed to provide functional evidence and publish that this is a likely PCD causing gene. So we showed that for that specific patient that their mutation was inherited in trans from both parents. The profile of this patient was that they fitted the PCD phenotype with a wet cough, bronchiectasis, rhinitis. They also had hearing issues and recurrent infections. The one thing that I would say is that this patient's electron microscopy actually looked fairly normal. There was a low-grade microtubule disarrangement, but this was the only feature that could be picked up. And one of the defining issues was that the high-speed video was highly abnormal with occasional long cilia. And CEP164 is a centriolar protein, and you can see in the control, you get a nice staining at the base of the cilia. But in the patient, this actually was missing. So the green CEP164 went missing from the gamma tubulin centriolar marker. And this was consistent in the ALI sample, so after we cultured the cells as well. So I guess we've demonstrated that this is a potential, so this is demonstrating how we've used our genomics data, and we've gone on to attempt to create evidence to uplift this gene as a potential PCD causing gene. But you can see it's not straightforward and requires a number of different tests. And there's still, there can be speculation about the high-speed video, speculation about the electron microscopy, which means that doing it on a repeated basis using your cultured samples to help reconfirm and as many different tests as you can is the right approach. You can't just, one test just can't fit all needs. So moving forward into transcriptomic analysis, we're now using RNA extracted from airway epithelial cells, and we're trying to specifically look for splicing variants. So Splicing is a normal process to eliminate introns from the DNA sequence in order to make an mRNA that is the template for protein translation. But there are a number of different instances where you can have aberrant splicing, where you can get exon skipping, you can get a mutually exclusive exon, meaning that you get two different products. You can get retained introns, which lead to completely abnormal protein length, or it can disrupt normal protein translation. You can also get alternative promoters, changing the way that the protein is made, alternate terminations, which mean that the proteins are truncated, and alternate donor and acceptor sites, which means that you get whole deletions, whole deleted sections of DNA. So a student who just recently gained his PhD just before Christmas, and this is work that's in submission to the ERJ at the moment, has developed a manual pipeline for transcriptomic analysis. So taking a nasal brushing, and we can either do this directly into RNA later, which is a way to harvest and safeguard the RNA until analysis, or culturing the cells. Sorry, I don't know if you can see my cursor. Culturing the cells at ALI and extracting them at specific time points has allowed us to confirm that we can successfully acquire and quantify RNA that can be sequenced. We use a deep sequencing method using an Illumina platform. So it's a short read platform, but it's deep sequencing. And then you can take the data and start processing it through alignment softwares, so that you can align to the genome and then start to interrogate whether you've got differential gene expression or alternate splicing. So there's Edjar, Salmon, and Armats and Majik are Yelmer's preferred tools for this. So this manual pipeline, I would say, is not for the faint-hearted. It's really labor intensive, and the functional evidence that you need to accrue to build back into the analysis, you need to be able to compare the data with nitric oxide, high-speed video, and EM to basically see if it fits what the analysis is demonstrating. And in the future, we're looking at using an automated variant predictor tool that will streamline this system faster. So this is just to give you one example of a patient that came for RNA-seq. So as an example, they had a single pathogenic frameshift variant in CCDC40. They had low nitric oxide, they had dyskinetic cilia, but their standard panel of their standard IF panel was normal, which included radial spoke, protein detection, and dining arm detection. And so this was slightly confounding. But they did have a hallmark class 1 microtubule disarrangement with inner dining arm, and I'll show you the ciliary function was extremely abnormal, as you can see along here. So we took the sequence and uploaded it into Congenica, which is a resource that can search for variants. There were no loss of function variants or variants with ClinVar pathogenic flags. There were no variants identified after extending to 124 gene ciliopathy panel, which is much more expensive than just the PCD panel. And there were no intragenic copy number variants considered, and SNP array didn't reveal a second mutation. So this was a perfect case to go into RNA-seq analysis. So we grew the cells up and harvested them at day 21. And Yelmer put them through his manual pipeline, and he demonstrated, so you can see on this sashimi plot that the control between exon 9 and 10, this is a normal splicing event here for CCDC40. But the patient had 2 splice patterns. So you can see the normal one, the normal one underneath, and then you can see this abnormal pattern on the surface. And this was due to a pseudo exon. So this was a highly cryptic, deep intronic splice acceptor site variant, and it caused a frame shift with a premature termination codon. So the argument was that this, in fact, was the second pathogenic variant. So the problem is that this variant was intronic, meaning that you couldn't pick it up by anything other than whole genome sequencing. So you won't find it through panel screening, or you won't find it through whole exome screening. But you could potentially go back and Sanger sequence to confirm the variant in the genomic DNA. So in order to satisfy American College of Medical Genetics variant classifications, there are different, so this is basically a classification tool. So if you have a variant, you need to go to this classification system, and it's a way to classify a variant as likely pathogenic with a certain amount of certainty. But you might need to go back and prove that the parents also have at least one of those mutations in their genome. So the problem here arises for this particular patient is that at present RNA studies need to be reviewed so that experts can agree the level in which this part of the classification can be applied. So although we can do this analysis, further work is needed to uplift the functional relevance of this data going into variant classification, which will help us in the future to be able to better clarify which variants patients have, but spot them more readily and report them more appropriately. So what about curative molecular therapies? So far, restoration of ciliary structure and function isn't yet achieved in vitro or in vivo. So what's been tried? In 2009, there was an approach to use lentiviral vectors to transduce an autadine arm gene, DNAi1, cDNA, into human cultured cells, which showed that autadine arm structures were increased when looked at by TEM, and there was some ciliary motility retained, but the efficacy wasn't very good. And the problem with most PCD genes is they're absolutely huge. And so a whole gene fixing approach isn't particularly feasible. And if efficiency of uptake of that gene incorporation is low, then it means that this isn't particularly going to be a go-to therapy for most patients. So what we're more focused on is looking at how we can edit genes to overcome the issue of whole gene replacement. And in order to do that, we need to disrupt or repair or replace sections of the variant genome. So in order to achieve this, the technology needs to have site-specific double-stranded breaks. You need to be able to engineer nucleases and cause a homologous recombination of DNA segments to rectify the mutation. And one of the approaches to do this is using CRISPR-Cas9. There are other nucleases that have been tried that have a lower binding affinity, I would say, and CRISPR-Cas9 at the moment is considered to have less off-target effects. But there's a paper by Lay et al. That demonstrated partial restoration of DNH11. But because of off-target effects. This is something that's holding this technology back. What you don't want to do is you don't want to try and engineer defective DNA in a patient, but actually cause them other deleterious problems. So some of the things that have hindered work of new curative therapies is the fact that ALI cells are really resistant to treatments, so particularly to viral vectors. So at the moment, we're looking at alternatives to viral vectors, so virus-like particles or lipid nanoparticles. And there are a couple of experiments that have taken place where lipid nanoparticles delivered intranasally to mice demonstrated, although they only showed by Western blotting and immunohistochemistry staining, that they could get an increase in DNA I1 expression in these cells. But this isn't yet transferring into patient samples or patients. And so this is yet to be taken forward. Another potential is read-through drugs. So if you have certain variants, splice variants that you want to try exon skipping, there are certain drugs that have been approved for use in other diseases. But to date, we've not seen restoration. So there was one attempt to restore Makaidas gene function by Lee et al. But the cilia didn't regrow on use of read-through drugs. So what does the future promise? So at the moment, I can tell you that Haymat's Omran is attempting to use messenger RNA treatments for CCDC40 patients. So he's, in brief, he's used patients with biallelic pathogenic homozygous or heterozygous variants that are proven, and then he's taken their ALI cultures and he's treated them with a repairing section of RNA on a lipid nanoparticle. So what he has demonstrated, what he's presented to us so far is that you can get some partial and slightly variable restoration of CCDC39, GAS8 and DNA LI1. So this is for the inner dynin arm, and these are parts of the nexin dynin regulatory complex and the ruler proteins that govern disorganization of microtubules. So he sees partial restoration of these, but that we don't yet have any evidence that ultrastructure is repaired or that ciliary function is repaired either. So although he saw partial increases in ciliary beat frequency, we're not yet seeing that the cilia have truly been repaired. And I believe that this is work that is coming forward, and we are to watch this space. So other nucleic acid therapies, there are RNA interference therapies, post-transcriptional disease gene silencing therapies. So you can use short sections of RNA and or DNA. So what we can do is use something called, we can use these azotherapies, which are internalized into the cell. They internalize using endosomal and lysosomal pathways. They enter the nucleus, they hybridize, and they prevent certain sections of mRNA translation. And this will be really good for potentially altering aberrant splicing. So if you can prevent that translation, then you permit normal gene function. And so this has real promise going forward, as well as the other mRNA strategy. But as I say, restoration of ciliary structure and function isn't yet achieved. And this is something that we'd really like to move forward with, because our patient communities are telling us that this is what they want us to work on. So for future grant Funding, this is something that you've really got to consider, speaking to your patients, charities, people linked to giving you feedback on the quality of your research question. But it's certainly one at the forefront of the UK PCD Support UK. So in summary, better genetic profiling and being able to upgrade variants of uncertain pathogenicity will help us to better stratify our patients. And you saw from what I showed you with the Geno-Pheno study, with just those 3 genes, it allowed us to significantly understand which patients were more likely to have heavier symptoms requiring more aggressive treatment, and which patients were more likely to be missed through phenotype alone. We hope that better genetic profiling will help us to detect patients faster. So at the moment, the UK median age of diagnosis is about 5, but as I've told you already, patients as young as 2 have bronchiectasis. So this is something we want to be better at. Finding new variants is really important to us, because we want to close the gap on that 30% that we don't yet detect using standard genomics. But also being able to classify these variants. It's no good being able to spot variants if we can't actually classify them as being pathogenic or not. And we want to be better at our diagnostics. We want to improve our management guidelines and tailor our management to patient groups. And by understanding what the genetic profile of our patients is on a whole, and better understanding the prevalence of PCD, which will help us to gain our funding, it will help us to target molecular therapies to create some advancements. So this is what the community is striving towards at the moment. So in my final page is to just acknowledge the fantastic team at Southampton, but also the wider national service in the UK. They're a really lovely bunch of people to work with. We coordinate with each other all the time, and we liaise with each other to make sure that each other is performing to the best standard possible. And I would encourage anyone out there that's feeling isolated to reach out to other centres, and you're welcome to reach out to the UK centres for support with your diagnostic training. And I'd be happy to take any questions at this point. I'm not sure if I've overrun.
Thank you, Claire. Amazing.
How do I stop sharing? I'll stop. Shall I stop sharing? Shall I keep them on my screen? I need to switch my light on. It's gone dark. There we go.
You can keep your slides as they are.
Yeah, and then if anyone wants to go back to anything.
Yes, I think that's a good idea.
So if anyone has any questions, if you'd like to pop them in the chat, but I'll start off with one of mine. So moving forward, looking at molecular therapies, what would be, in your opinion, the best way to model? So if you didn't have access to primary human epithelial cells, for example.
So at the moment, I'm attempting to look at use of a cell line. We're very limited with what cell lines are available to work alongside. So there are very few cell lines that people have utilised in the past that have actually been successful at regrowing their cilia when you try to culture them. So there are a couple at the moment that others have used. So the BMI1 cultures that Chris O'Callaghan's group use. There's also a cell line produced by Cornell. But I think with the lack of cell line resources available to modify and study PCD at the moment, our best strategy is to coordinate with other centres, because this is still a rare disease and our own bioresources are limited to our own gene pools, reaching out to other networks and not working in a silo is probably your best way of improving your ability to use primary samples as a first line. Some people use spheroid cultures, but I personally prefer the ALI platform because I think you get a more representative pseudo-stratified epithelium and mucus production. And so I think primary cultures are an excellent starting point for the bench. I think mouse models, we should probably be aiming to reduce them if possible, but I know that there are talented geneticists that are looking at CRISPR editing mouse models for PCD cure development. So I think that you have to go with the strength at the center that you're working from and try and network as much as possible. Sorry if that's a bit of a nebulous answer, but it's my own approach.
No, thank you. And collaboration is key.
Yeah, thank you very much.
Everyone's gone to sleep.
No, certainly not. There's still a lot of people here. Thank you very much for your excellent presentation, guiding us through those very lively videos and images of PCD diagnostics. I have one question. Now, when you are actually looking at, for example, immunofluorescence or any kind of proteins that you think are actually hiding, but are not they are not stained and visible, but you think there's something out there that would actually probably also connect or explain some variants of unknown significance. Do you sometimes encounter that when you feel like, oh, there's just this tool or this step missing? What would it be?
So for instance, we've got lots of example of the dynein assembly factor variant. So we've got a variant of uncertain pathogenicity in an assembly factor. And if you look at the IF antibodies available for those DNA-AAF assembly factors, for example, they have variable localization, and they don't always stain the cilia even in healthy samples. So you can't use it as a reliable go-to staining method to demonstrate a certain specific loss of protein if you can't prove that it's always present in the healthy control. So we've got lots of cases where we've got these assembly factor variants that we can't really perform immunofluorescence to help prove a specific protein has gone missing. But as I also said, the dynein arms, for example, are built up of hundreds of different proteins that, you know, if you and they assemble in a sort of a chain of events. So if you lose one particular protein, it might mean that you lose the whole structure. So which one caused the defects? And there's not very many specific tests that you can perform to nail that. So what you have to do is take perhaps a more pragmatic view. You have to look at what functional evidence you have, and then you have to see if it fits the profile of the genetic variants that have been considered as the cause of that person's PCD. But there are initiatives in place for collecting PCD variants. I'm thinking of a particular project that I won't mention because it's not out there yet, but there will hopefully in the future be a database that allows us to search for specific PCD variants, which will mean it's easier for us to say, well, we've seen this particular variant in this patient, but somebody else somewhere else in the world has also detected a patient with the same variant, and they have the same clinical features and same diagnostic profile. So this will help us in the future.
Very much clear.
So I've got another question. So moving forward to a time where perhaps the RNA splice variant process is automated, what kind of input would be required? And would this be straight after nasal brushing, or would this be after expansion of the cultures?
So I'm hoping that we'll be able to go straight from nasal brushing, that we won't have to spend time culturing the sample, and that we'll be able to detect all of the potential splicing variants that we're wishing to detect. So we've already started so Yelmer's projects, which hopefully will go into the ERJ hopefully soon, is demonstrating that you can definitely reconfirm when you have a pathogenic variant, and then you can show some difficult cases that have splicing variants proven by his transcriptomic analysis. So there'll be a period of validation required from an automated method, and this is what we strive for in the future. So we'll validate on a subset that we have the data for, and then take it forward into unknown cases. And I think that's the right way forward for that, to prove that we can definitely brush a patient and then get an answer at the other end without having to culture them. Cultures are a backstop.
Thank you.
Is there anybody out there, if I might ask a little question to the room, and I don't mind if nobody replies, but is there anybody out there that is seeking some support to develop skills in diagnostics? Or am I speaking to an audience perhaps that has been already doing it for a while, and you have specific go-to tests that you're not sure about? Is there anything I can help with? Nothing.
No one in the chat and no raised hands so far. Okay, maybe I continue with another question then. Yeah, you just mentioned cell cultures with the ALI cultures, and so retesting certainly is reasonable, especially in people that are suspected of PCD, but so far just with none of the diagnostic methods, PCD was proven. And so those cell cultures came in very handy. Now, speaking of genetics, do you already maybe take blood samples and then keep some of those in reserves to then have some kind of culture, not culture, of course, but those reserves to repeat PCR testing and test for new genes that are added to the PCD list actually annually, or how do you do that?
Yes, so what we've had is patients in the past, they've had genetic testing under a 29 panel, let's say, and then new panels have been updated. And so we've reviewed the patients at a genetics MDT, where we keep them on a list of unresolved cases. And sometimes we've gone on to then repeat with the 46 panel, which is the expanded panel. So I guess we do keep the genetic material available to repeat tests, and obviously send for whole exome sequencing. But I guess the question for us now is, where is the place that transcriptomic analysis would fit? And yes, we store cells from nasal brushing so we can regrow them at a later date. So we use a product called, other products are available, but we use CryoStore because we trust that it keeps the integrity of the cells quite nicely. You can freeze the cells down at what we call passage. It's in the Coles paper that I cited during the presentation, but we freeze the cells down and we can bring them straight back out onto transwells. So you can actually fast track a little bit, in that you just have to wait for them to ciliate before you harvest and repeat tests. So you can store cells, you can store blood, You don't have to do genetics straight away, or you can send for genetics or repeat genetics. We try and think of everything, so we don't have to recall the patients.
Thank you.
Thank you very
A
much.
question from Nuria. Wonderful talk, Claire, thanks. We've always had trouble in extracting RNA from samples. Do you recommend RNA later and extract in 24 hours, or are there other methods that you've used?
So sorry, I'm just going to read that. So we've always had trouble extracting RNA from samples. So how much are you starting with? So RNA later. So normally, if you put your cells into RNA later, there's usually a refrigeration step before freezing if you want to store them longer than extracting them straight away. I know RNA later is stable at room temperature, but if you want to long-term store your samples, you could do to acclimatize them before freezing. So I presume Yelmer's paper will have the method that he specifically used. And the only other thing I would say is you need to work out whether you've got enough of your starting cells to extract a reasonable quantity of RNA. So what did he use? I think we went from one large transwell, so a 12-well format, and that was enough RNA to onward RNA-seq. So I think, yeah, we didn't always extract straight away. Sometimes he extracted fresh, but we also had cases, particularly during COVID, where we had to freeze the samples and he extracted them later. So we've proven we can do both. I can see Amelia's made a comment. We'll be announcing. Oh,
No, I
does
think
Amelia want to say something?
a question is down below. If you were setting up with limited resources, what tests
Yeah.
would you include?
So with limited resources, so if you don't have access to electron microscopy, you could perhaps, because we've got partners from the partner springing to mind sends us samples in fixative for us to do electron microscopy screening for them. I shouldn't call it screening, testing for them, and then in their own centre, they've managed to accrue a nitric oxide gas analyzer, and obviously you can perform the Picadar score for free. If you don't have access to nitric oxide screening, I think at the very least the Picadar score, and then attempting to get some electron microscopy screening. But the IF could be potentially a cheaper way, but you need to have a microscope that will allow you to image your fluorescent staining, and then you need to be able to have that sense of not hanging off the first result. You might need to repeat to make sure that it wasn't just a control error or something like that. So there is a paper that Nisreen Ruman wrote a couple of years ago talking about her experience as a limited resource setting, and I would encourage anyone to go and have a read of that. So I think there's a DNH11 question.
Yes, so a question from Iwa. Have a question concerning DNAH11. In our experience in Poland, the majority of mutations seen in this gene are amino acid substitutions. Do you have a similar pattern in your population?
So you mean the majority are missense mutations rather than nonsense? I'm not sure off the top of my head. I'll have to go and have a look at that. I'm not sure if I can answer that question accurately. I don't think that, I think the good thing about DNH11 cases is that if they have a loss of DNH11 staining and your EM is demonstrating that the outer arm structure is present, it can increase your confidence of maybe an amino acid substitution mutation. I think because DNH11 antibody labeling can also be falsely present, that's the only caveat, that they can be still a bit tricky to diagnose. But that's the only thing I would say, that if you've got a normal ultrastructure and you can see that the function is abnormal and you have a loss of DNH11 antibody labeling, I think that's quite a strong indicator that your mutation is pathogenic. I would encourage you also to speak to a geneticist who would agree with that, putting that evidence together as well. So definitely have difficult case discussions if you can, and hook up with other centres for difficult case discussions. Use the BEAT-PCD difficult case discussion.
Great. So with no more questions, I'd like to thank you, Claire, for an absolutely amazing talk and this really vivid discussion session.